So Simon studies egg and flowering time in bread wheat and the genes that control the interaction between these traits to influence yield. His research undertakes large-scale screenings of wheat lines using mapping techniques to identify QTL for traits of interest. He uses field phenomics platforms to measure traits in real time under agricultural conditions and identifies markers targeted to specific environments. And these are used to inform breeders for traits of interest and for doing pre-breeding germplasm. So Simon, thanks for being here. Okay. Would you like Shal to? It's okay, better? Okay.
Let's come from the clip. Thanks for welcoming me to Pergamino. I've had a fantastic trip. My first trip to Argentina so far, with Gustavo Schleifer and Daniel Moraches hosting us from the University of Buenos Aires. It's a shock that I've never been here before, because we've collaborated for a long time with groups from Argentina, and I really can't say how much of a pleasure it is to be here today. I'm going to roughly talk about three things. Fernanda said I'm interested in adaptation and breeding, so I'm going to talk on the science side about those two things. But first, I just thought I'd tell you a little bit about the John Innes Center, which is the research institute that I come from. So the John Innes Horticultural Institute was opened in 1910 in South London. And then it moved to a place called Bayfordbury, north of London in Hertfordshire in the late 40s. And in 1960 it became the John Innes Institute. It's now based in Norwich. That began in 1967, and this is my office here in something called the Cambridge Laboratory. And it's called the Cambridge Laboratory because actually the people I form my main groups with at the John Innes Centre, where we came from is the Plant Breeding Institute in Cambridge. So this, after the privatisation of plant breeding in the UK in the late 1980s, it was a right-wing policy of Margaret Thatcher to privatise the public breeding programme. And the research, the private part became companies like REGT and Unilever and Monsanto. And then the more fundamental research part moved to the John Innes Institute in 1990. And then the merger of those two major UK centres became the John Innes Centre. Please feel free to get in touch with me. And if you're ever in the UK, please do come and visit. We'd love to have you in Norwich. One of the disadvantages is we're in the east and we're at the end of the railway line, so people don't come to visit. But it'd be lovely to see you. Okay, so the breakdown of John, it's up to 1,000 staff members, but half of those people are support staff. So they're maintaining the building, the services within it. We're very well served in that way. But half of us are scientists. So this is 46% kind of permanent scientists, including 50 project leaders, which is what I am, so a principal investigator. 15% visiting workers, 22% postgraduate students, and 17% postdoctoral. A major part of our mission statement is training, but the fundamental thing, the kind of strapline for the John Innes Centre is genetics. So we're about the genetics of plant and microbial organisms. We sit within a wider research park, so we were just discussing the kind of the politics in Argentina of what other institutions you're associated with, and one part of the success of the John Innes Centre is the association with other institutions. So the building you just saw is here, the Cambridge Laboratory. This is the Sainsbury Laboratory. So they're funded by Gatsby and the Sainsbury Trust, and they focus on pathology. This is the Earlham Institute, so this is another institution funded by the BBSRC working on genomics. This is a new building called the Quadrum Institute, and they come from the Institute of Food Research. Here, outside the picture, is the main regional hospital, the Norfolk and Norwich Hospital. And the helicopter that we're on at the moment is flying over the University of East Anglia. So these interactions are very beneficial for us in many ways. Okay, so getting on to the adaptation. I did my PhD with Dave Laurie, and we were cloning genes for heading days, but became increasingly interested in what happens either side of heading days. In terms of crop development, how does this influence performance? And people like Fernanda and Gustavo were very influential in us thinking differently about why phenology is important. So I'm really showing you this just because it's a bit of fun, I suppose. I work on phenomics as well, and a colleague of mine called G. Joe makes a static camera that films the crop through development. We use machine learning to recognize things within the canopy. So these little red dots are placed there they're not, we haven't drawn them on, it's the computer recognizing what it's calling the top of the canopy. But I think I like the way it bows at the end. But the point here, the simple point, is that floral induction already occurred when we started this film. So that happens in the UK in March. We drill the wheat in, we sow the wheat, usually in September. Has that winter of building up biomass, and then it's made the transition from making leaves to making a floral meristem by March, so just after winter. And then the whole process after that, so everything we've just seen on the film, is to do with yield formation is occurring through inflorescence development, and all of the environmental factors and genetic factors that can influence yield are strongly concentrated within this part of the crop growth cycle. So we wanted to start to study in more detail that part of the growth cycle. Okay, so our justification for doing that and investing resource in that would be something along these lines. So this is I think a mental model that many people have about adaptation, where what you need to do is hit this sweet spot where the dangers of the winter are sufficiently past for these delicate stages of development, meiosis, booting, and thesis for the crop. But then not so late that you're moving grain filling into heat stress and drought stress in this region. Or in an environment like the UK, you're just making it too late to combine the crop. And I think an assumption that many people made was, so that's what I meant to do there. Avoid those things that we don't want to hit. And an assumption that was made by many people working on phenology was that anthesis date or heading date was a sufficiently good proxy for those other parts of development, that as long as you have anthesis date in the correct target date, then things like grain filling, meiosis and booting are also going to be roughly in the right place. So we did some experiments to test that. This is a very elite cross. This is between two UK feed wheats called Buster and Charger that flower at exactly the same time. They're fixed for all the major phenology genes. So this isn't about, big flowering windows and mega environmental adaptation. It's exactly the kind of variation that UK breeders use throughout their program. And we found that within this cross, there are two heading date QTL, very consistent. This is on our experimental farm near John Innes. Over 3 years, we pick up these QTL on chromosome 4A and 2D, and they're the genes controlling heading days in the Buster times Charger cross. But if you go back to floral induction and say, well, something closer to that is terminal spikelet, the very last thing the developing inflorescence does before it begins to extend, it makes this last spikelet, which is like a tap turning at the top of the wheat ear. And again, very clear, consistent QTL. is on chromosome 7A. I hope you can see it. But nothing at all on 2D and 4A. So actually, the two processes are completely independent. And the idea that anthesis can be used as a proxy for anything beforehand is mistaken. This was work done by Oscar Gonzalez Navarro from Mexico. And although I was a supervisor, it was part of an adapter wheat project that Gustavo and Fernanda helped me with. Okay, so we're interested in these things, and we're interested in the environmental variables that the crop has to be adapted to or to use to exploit to produce a better performance. And this is the UK season, and this is data from our Met station. So as I said, we sow the seeds in September and we harvest in August. And then the main photoperiod and temperature signals that crop can use are shown here. So here in the UK we have rain, which is no kind of signal at all, completely volatile and variable thing. Then temperature, and actually temperature is much more of a reliable signal. If we look more closely at that, this would be a much more jagged line than the photoperiod, but still I think temperature is something we can use. It almost looks like the growing crop canopy. But then photoperiod is a really steady signal, where you see this variation in photoperiod. In the UK, wheat is a photoperiod sensitive plant, so it's waiting for those long days before you get full heading. But if you actually look at the photoperiod during this period of growth, and I don't know, if we call 12 hours a short day for this long day plant, then you can see that the vast majority of the development I've talked about actually occurs under short days. It's not under long days. And floral induction, that terminal spikelet stage that I talked about, that happened under short days. That was in March. So here's March. And the day length in March is just getting into 12 and a half hours. So when people talked about the importance of photoperiod for wheat, they often talked about PPD1 and photoperiod insensitivity in long days. But I began to wonder how important are short days for changing the performance of the crop ultimately. So we began to, the way we always do this is just like a mutation screen, but instead of using a mutant population, we use elite germplasm. And we saw that most of the UK wheats we screened this kind of behavior, where this is a short day, very delayed flowering after the 1st of May, so this is really late. Then you give it a long day, it becomes a lot earlier. And even when you extend that day to a 20-hour day, you get a little bit of extension in heading date. But you get some interesting exceptions, and as geneticists, this is what we look for to map the genes which are important. And you can see here that Rialto, again, photoperiod sensitive UK winter wheat, is really not so late as the others on the short days. It's got something special. So we made crosses with these things. It's become ridiculous how many biparental populations we've made now. So we got tens of thousands of recombinant inbred lines that we dip into to map these things. And in this case, we're looking for a short day specific QTL. So this is the gene that's controlling this thing. And sure enough, we see it on chromosome 1B in a cross between Spark and Rialto. So Spark had the normal behavior, Rialto had this not so delayed flowering. Reasonable QTL under short day. Short day experiments are difficult because the plants are sick. So actually the variation you get is strong, so you get quite weak QTL. But there's nothing under log days and a clear significant QTL under short days. This turns out to be a gene called, so we fine map the effect, and this is a gene called FT3, which is actually in barley already known, so this is PPD2. And the way it works is that it promotes flowering under short days, so the presence of FT3, if the summer's coming and spring is passing, and still because there's some vernalization requirement that isn't satisfied because of the nature of the winter or the season, it's genes like FT3 that override that and say, well, it's time to flower, just go. And in Rialto, you see this mutation in this very conserved domain of the protein. It's a phosphoenol pyruvate binding region. And in here, in Spark, it's a serine. And in the rest, it's a glycine. So it's this polymorphism which is responsible for changing that photoperiod response. As I say, we want to have impact in breeding, and the way we always do this is to make the markers. So in this case, this gene FT3, we worked with CIMET to see if it was important in their material. And that's a CASP marker based on that one amino acid change I showed you. And there's a few things about this. First of all, it was great to see there's a positive association from a little field in the east of England to this association panel in Obregon. It's still important. But also about association analysis. If you look, there's no collar. There's none of these markers in the high density array are associated with flowering time. This is to do with the marker distribution and linkage disequilibrium, and it's just a lesson to me that association genetics works, but not always, and you really do have to be careful about interpreting this data. Only by making the perfect marker based on the functional polymorphism did we see that gene. So I don't want to go over too many of these examples. We've mapped a lot of genes now, but if we skip to the next one, just because this became an interesting story about that other part of the meteorological data, the temperature part. So this is, that's FT3 on chromosome 1b. But we did exactly the same thing for an alienus per se gene. And alienus per se is all the variation that's left after you've completely satisfied vernalization and you've completely satisfied photoperiod. Again, we made a nice marker. This time the I-select markers worked well too, but our marker is the very top one on there because it's the gene. And again, for CIMET, this was useful because it allowed them to understand their variation in phenology. But we just characterized it as alienus per se, which is this dustbin category for things that aren't photoperiod or vernalization. This work was with Sivakumar Sukumaran and Matthew Reynolds at CIMET. But the reason the link with the physiologist is really interesting and useful for us is because they can tell us a bit more about what's actually going on with these effects. And this was work that Elga Ochagavia and Gustavo's team did in Lleida, where we made near-isogenic lines for the 1D effect, the EPS. Everything we do, we make isogenics for pre-breeding and for gene discovery. Because you can't clone QTL, you can only clone major genes. And what we found is that Gustavo had the hypothesis that many EPS genes are likely to be involved with the interaction with temperature. And by growing these materials at 6, 9, 12, 15, 18, 21, and some higher temperatures, 24, I think, degrees centigrade, you can see that there's a very clear interaction where at the low temperatures, the difference in flowering time with genes, with lines only carrying these alleles is higher. This is the difference in flowering. And as temperature increases, the difference changes. They're still significantly different, but they become very much closer to each other. And then at 21, something crazy happens where actually none of the late flowering alleles lines flower at all. And then at 24, nothing flowered. So by doing this experiment, what was a small flowering time effect for us, 5 days difference, Gustavo's team had turned into what was approaching a lethal mutation by looking at that range of temperatures. So I think there's a few messages here. First of all, the EPS category probably is in many cases about temperature interaction. And secondly, by looking at the same gene from different angles, you can interpret them in very different ways. Okay, this is down here is the ones that never flowered at all, and this is the example where, in most cases, the later allele just didn't make it through. So you can see the early ones and the late. This is an ongoing process, so although I don't really understand how to put these things together in breeding yet, or even in some kind of model for how they all work, we think it's important that we start to identify the genes controlling short day response, because it seems to be a very rich vein of undiscovered genetic variation. This is the latest one I've done, where again we 3 day lengths. In this case, all what you would call approaching short days, so 16 hours, classically you might call a long day, then intermediate 13, and then 10 hours. For most, you see this kind of expected response, late, a bit earlier, a lot earlier. But you see some interesting ones. These are all from Scandinavia. And pretty much all the Scandinavian material is fixed for this genetic variation where a 13-hour day looks like a short day. It doesn't make it any earlier by increasing the day length to 13 hours. Again, we mapped that, and there's a clear single QTL on chromosome 2b. I was worried it might be just an allele of PPD1, which is the famous photoperiod-insensitive gene, but that's here. So there's something here, a gene which we think affects the critical photoperiod or the threshold photoperiod for what the plant sees as a short day. It changes it. So I'm really excited by this one. And you can see here, we've had a step change in wheat where the genetic maps are so dense you can hardly see the markers anymore. The wheat genome was fully published with annotation in July of this year. So it's going to be like rice. We clone these genes very quickly now. What used to be an entire postdoc, if not more, years of people's careers, is much quicker, and we're going to clone these genes and be able to give the perfect markers for breeding. From a breeding point of view, the way I'm thinking of trying to apply this knowledge is through drilling date. This is drilling date in the UK, when it was drilled in September or before the 20th of September. It's data from like centralized data collected by DEFRA in the UK, the government department. And you can see that in the 1970s, nobody drilled in September. All drilling was done in October and November. And then there was a trend which is very closely correlated with the trend for yield increase in the UK, where people did drill in September, and they just get earlier and earlier to the point where then from 94 onwards, a lot of farmers are drilling before the 20th. So now it's very much, we combine the wheat in early August if the conditions allow, and people get ready for drilling immediately. So the wheat's grown, in many cases, for a year. The exact reasons why this is important, etc., we can discuss. But what I can say is for the genetics of the crop, this is really challenging because, in September it's very warm, the day length is long enough. The crop can, if you place spring wheat at that point, it'll proceed and flower by Christmas, and you'll have some seeds on it. And then the things which do well are really surprising. So you might say, well, all you need is a really strong winter requirement, stop and wait for winter. But the most flexible variety in the UK is a biscuit wheat called Clare. And it only has 2 weeks vernalization requirement that you could easily satisfy when it starts to cool down in October and November. But actually, Clare, you can drill from the very earliest date, just after combining, right through to almost like a spring wheat. So there's something other than vernalization which is important for regulating this response. And I think it might be some of these genes. So the way we're trying to use them is to understand drilling flexibility. And of course, all farmers would prefer to have that than that. This is called the recommended safe drilling window.
What is the dotted line and the straight line?
The dotted line, these are based on drilling dates by our recommending organization, the AHDB. This is the safe drilling window. I think this is, you can risk it. Actually, I'm not. That is the safe drilling window, Gustavo. I can't remember what that is now. Sorry. Yeah. I'll try and work it out. Okay, but this is good. I think we can help breeders with this information, and we do interact with all the major commercial breeders in the UK. But the thing that we all found frustrating about this way of working is that all the genetic variation I've described to you is already present in the gene pool. So it's already there. The breeders have it. Hopefully we can help them to shuffle it better with molecular markers, or just by knowing it's there. But it doesn't really allow us to say, well, this is a desirable phenotype. If we want to extend it even more, if we want to do that, how do we get new genetic variation? How do we introduce that into the elite breeding pool in the UK so that we can extend phenotypes which are desirable? If you can't do that just by shuffling the existing pack. Okay, so a whole new program was established in the UK to do this. This was set up in 2011. We have a 5-year funding cycle, and the current funding cycle is called Designing Future Wheat. And it's based on exploiting natural variation. So this is the evolution of bread wheat. This polyploidization event to make pasta wheat happened maybe 500,000 years ago. The polyploidization between pasta wheat and this goat grass happened 10,000 years ago. The global spread of those cultivars, those landrace cultivars, produced landraces, so it means that locally adapted types were adopted in various environments, and then without systematic selection they became better for those environments, so they're called landraces. Mostly in the kind of mid-20th century, systematic breeding began, and we start taking landraces, shuffling the pack, and making even better, fine-tuned varieties. So the question here, the fundamental question is, when we took this decision to enter systematic plant breeding, did we leave behind some useful variation in this? Different groups took charge of those areas. So at the University of Nottingham, they look at wild relatives, the most challenging and difficult part of it. So you're going right back to wheat relatives, crossing them with wheat, and breaking sexual and meiotic recombination barriers to introduce new chromosome segments. At NIAB, they copied the CIMMET approach for like Mujib Karzai, made new synthetics, where you cross a durum wheat with a goat grass again, and you reinvent that event that happened 10,000 years ago, but with different Tauschiais. And at the John Innes Centre, I led the landrace pillar. So that was about all these landraces that spread around the globe, what's in them, what's useful for breeding. The whole project with Graham Moore's idea, so he leads the UK wheat programme, and it was strongly supported by the commercial breeders of the time, who are referred to as Pillar 4. So if you think about it, we're looking at the wild germplasm, but the elite germplasm is something in itself, and they're doing the fine-tuning. The genotyping's at the University of Bristol, and the phenotyping at University of Nottingham and Rothamsted, led by Rothamsted. I'm going to talk about the land races because that's my thing. We had one beautiful collection called the A.E. Watkins Landrace Collection. So in the 1920s, contemporary with Vavilov in the Soviet Union, Watkins just took a very simple measure. He sent a letter out to all the British embassies and he said, go to the local markets, buy some seed and send it to me. And very interestingly, we've got the original letters, and he said, under no account go to the local breeding stations, because what he didn't want was improved material. He wanted them to go to the more remote places. He knew that systematic breeding was starting, and this was a chance to take a snapshot of global diversity. And it's quite incredible in the letters how he spelt it out. That's not just me kind of fantasizing about how he might have thought of things. It was very clear in the letters. This was done to make a collection of 100,000 cultivars. Unfortunately, over time they weren't looked after well, especially during the Second World War, and that's down now to 827 original landrace cultivars of bread wheat. There's also a durum collection. But the 827 is still an unbiased selection from all of these countries, and the size of the circle gives you an indication of the number of lines. Where you see the lines from the former Soviet Union, they were provided by Vavilov to Watkins, and they exchanged material. Okay, so I need a strategy to exploit this material. And again, we tried to think in a physiological way about how that might work. This is grain yield at 2 nitrogen concentrations at Rothamsted and the University of Nottingham, using the Watkins in purple, the synthetics from NIAB, the new synthetics in green, and the current elite varieties in the UK in blue. And you can see very clearly, and it shouldn't be a surprise to most people, that the landraces perform really badly. They're low yielding. There's no point in trying to identify useful yield component traits directly in the landrace. It's just not there. As a kind of clue to why that is happening, you can see that the landrace material is really tall. So even with 4 applications of plant growth regulator, the tallest landraces are taller than me. Without plant growth regulator, they just lodge immediately. So it's kind of not a very hopeful story so far. But when you measure biomass, you can see that actually the landraces are making really promising levels of biomass accumulation. And the problem is that harvest index is just terrible. It's never been adjusted in the way that was done by systematic plant breeding in the Green Revolution. So it's not really appropriate in any way to look at them like a modern wheat. As a geneticist, we need another strategy. Oh, and finally, just from an agronomic point of view, it's impossible to work with this material. So with the PGRs, it's not really agronomically meaningful because they reliably lodge. Modern soils have residual nitrogen where you can not apply nitrogen for several years, and it's still too much compared to what these lines are actually adapted for. Okay, so we wanted to break the genomes up. We wanted to identify the parts of the genome which are most likely to not be in current breeding material, and then put them in current breeding material. So this is a structure analysis. It breaks up the landrace collection into an eastern. You see the color coding of the countries is there. And it means that the classic archaeological bifurcation of wheat to east and west is very clear. That's the eastern half, that's the western half. And then when you analyze them hierarchically, so you do the analysis only on that half, you get these ancestral groupings. And then if you do only on that half, you get these ancestral groupings. These make sense in terms of countries of origin, and they show that's our version of the structure of wheat before breeding. Okay. Keep doing it. Okay, so I said the Watkins collection was 827. Our collection is actually 1,050 because land races are not. Heterozygosity isn't the problem, it's heterogeneity. When people grew wheat, in fact, when land races are still grown, they are mixtures. So you can't do genetics on the landrace accession directly, you have to purify it. We purified the collection by single seed descent, and when we saw meaningful segregation in single seed descent, we made a new accession, which is why that's 1,050. Doing it, sorry. Then we wanted to make a manageable set for people to study. So we sampled each of these windows, the ancestral groups, to represent the variation seen within them and make a core set of 120 lines. So this is something that people could manage when they screened it for disease resistance or physiological traits or whatever. And it's also something we could manage in terms of making a genetic resource from it. To do that, we did it just the way I described. I wanted to do it in the way I understand, which is the way I clone those adaptation genes. And these are all the elite crosses we used to do that. But we just said, well, we'll do that for the land races. We'll make biparental populations from all of the core set. And then we can identify QTL, and then just like we do when we clone genes, we can make near isogenic lines, and we can move from this horrible landrace material to elite material just carrying the gene of interest. This is basically an adaptation of a maze strategy developed in the US. So it's called a nested association mapping panel, and it's what we did. We crossed everything with a UK spring wheat called Paragon to make a real 120 real populations of 94 individuals, which makes this, this large real set of over 12,000. This is the process. So this was single seed descent carried out by Simon Orford in my group. Very carefully done. So we make the crosses. We don't make the SSD in the field. We do it with bagged ears in the glass house, so it's very clean material up to F4. We only progress to F4 because that's clean enough, and it allows you to extract heterozygotes at the locus of interest. And then we multiply it up and grow it in the field. We made genetic maps for all of those individuals. These are CASP-based maps, and these are the CASP markers of the maps, a consensus map from all of them, aligned to the wheat genome. And then we also made the same populations from various target environments to try to benchmark landrace variation against mega-environmental adaptation. So lots of CIMMET populations, Kazakhstan, elite UK lines, Australian, Spain, and purely for molecular biology reasons, Chinese spring. This is just full of diversity that you wouldn't believe. The wheat genome is never like the Chinese spring wheat genome. This is 2-point linkage comparison of all the markers. Where you see these points off the line, it means there's a translocation. Translocations are the rule rather than the exception. And we show that they're real in collaboration with Nottingham by fluorescence in situ hybridization, where you see this translocation there. You find spontaneous aneuploids all the time, lots of translocations. All the material was phenotyped in work led by Malcolm Hawksford at Rothamsted and Nottingham. These are the experiments, a lot of phenotyping work. We do the QTL analysis, and we identify thousands of QTL in this material, so I can't describe them in any meaningful way, but this is a summary of the type of effects we have of the 1,272 QTL so far. We just use these as signals, and as long as they're not clearly negative, we backcross them. That's the backcrossing strategy. It's 2 backcrosses into Paragon. And then we've got this pipeline of near isogenic line development. So you're moving them from the real line to something which is completely like Paragon. The first set was just 11. They come from 2011, from the first QTL analysis. And every year they get more and more QTL. In fact, now we're up to set 8. And the team continues to backcross and backcross and backcross. The main reason for doing this isn't to clone genes or to do physiology or anything. It's a pre-breeding experiment. This is Simon Orford from our germplasm resource unit. He's talking to a group of breeders, academics, and funders. And he's basically selling this material. He's trying to flog it and say, look, it could be something that you're interested in. That's how we did it at first, and then the approach was successful. There was material that the breeders were very interested in, so we formalized it. This is called the Breeders Toolkit Committee. I chair it. Simon from the GRU kind of manages the approach. These are representatives of all the UK's commercial breeding companies. These are the work package leaders from Designing Future Wheat. Institutional representatives of my work package, the germplasm work package. Everyone here gets a vote, and every single QTL that gets backcrossed is accepted by this committee or not. Many get rejected. And actually anyone in the UK can nominate a trait of interest to be backcrossed into this pre-breeding program. Then, when the back lines are finished, they change from being breeders' toolkit